Good morning, good afternoon, or good evening, depending on where you are in the world. And welcome to today's Beckman Culture Microbiology webinar about the current topics and challenges in antimicrobial resistance. My name is Zakaria Hadha. I'm the Microbiology Products Manager for Middle East and Africa, and I will be your moderator for today. Before we begin, I have just a short announcement. You can see on the right side of your screen a small question icon. Please click on this icon to submit your questions at any time during the webinar. And now on today's webinar, current topics and challenges in antimicrobial resistance, our speaker will be Dr. Barbara Zimmer. So Dr. Zimmer is currently principal scientist, scientific affairs for Beckman Culture Microscan. She was a lecturer in mycology and infectious disease at the University of California, Davis School of Medicine in the Department of Dermatology for 30 years. Dr. Zimmer received her PhD from the same university, and she also works in research at Mayo Clinic. Dr. Barbara is the author of more than 30 papers and 130 posters, and has presented over 100 scientific talks and workshops. Dr. Zimmer was also a voting member at the CELISI subcommittee on antimicrobial susceptibility testing for 14 years, and now she's at the board of director in the same organization. Dr. Zimmer, over to you.
Hello, everyone, and greetings from California. I think it's one of the really great things. The pandemic, of course, is not a great thing, but one of the really good things that has come out of that is a lot of great telecommunications. So I'm able to talk to you today actually from my office at home. Thank you so much for coming to this discussion. I think discussing antibiotic resistance is really more relevant now at this time than ever before. So let me start. And again, if you have any questions, please feel free to type them in, and we'll try to answer everything that we have. Okay. If there's any audio-visual problems, please just let me know. I think, and I'm hoping that everyone can see a title screen, which what we are going to talk about today: antibiotic resistance, antibiotic stewardship, and actually some new antibiotics as well. So what we'll be doing today is talking about challenges that we have in today's microbiology laboratory that are not related to COVID, which of course has been a challenge to everyone worldwide, and particularly those in the microbiology lab as we start to test and struggle to test and add that to a regimen that was already overcrowded with a lot of things going on in microbiology. We'll take a look at some interesting facts about antibiotic resistance and the detection timeline. We'll know the importance of minimal inhibitory concentrations and susceptibility testing and why it's still relevant in a world today. We'll talk just a little bit about what antibiotic resistance is clinically important today, and we'll use the World Health Organization antibiotic resistance threats as a framework of that. We'll discuss carbapenem resistance, ESBLs, or extended spectrum beta-lactamases, colistin resistance, and methicillin-resistant Staph aureus. We'll talk a little bit about some new key antibiotics that are coming to the market, specifically those that are important in treating carbapenem-resistant organisms and ESBL-producing organisms. We'll talk a little bit about some basic concepts of antimicrobial stewardship in an era of bacterial resistance. We'll be using some examples from the US CDC, but of course those principles are appropriate to every place in the world today. And last but not least, we'll talk a little bit about Candida. We talk about antibiotic resistance in bacteria, but we these days have to talk about antibiotic resistance in yeast as well. So to start off just talking about our current challenges in microbiology, of course, we have to start at the top with COVID. It's affected not only our lives everywhere, but it's certainly affected our life in the microbiology laboratory today, as many things that we've wanted to do have gotten to be put aside while people bring on various types of COVID testing as well. We're being asked in the lab to perform more testing with greater volume and greater complexity. We're also coping with increasing shortages of trained microbiology technologists. This was true, of course, before COVID. There used to be a lot of people that could talk a lot about different aspects of mycology and different aspects of bacteriology. And a lot of those people are retiring. And we have to do this in an economic climate where reimbursement does not always keep pace with increasing costs or all the tests that are needed to be done on any particular isolates. On page 2, of course, I have to start with COVID again, since it's just taken over everything that we've done. But most importantly, we need to keep up with the emerging resistance in bacteria and accurately give susceptibility information about the etiologic agent of the patient's disease. We need to not only give SIR, susceptible, intermediate, or resistance, but MIC information when it's important to patient care. We need to keep up with changing antibiotics and changing breakpoints. And we'll talk about one today that's important, and that's with colistin. And finally, we need to incorporate rapid tests as appropriate to give identification and resistance information to physicians as quickly as possible. For example, methicillin-resistant Staph aureus. There are obviously a lot of new technologies that are coming into the field today. So what I thought we'd do is kind of start with a diagnostic microbiology timeline. And of course, it goes back to where animalcules or bacteria were very first described in 1677. It took another couple hundred years before we were able to culture bacteria. And that was followed very quickly by staining from both Paul Ehrlich for the acid fast stain for tuberculosis and Christian Graham for the Graham stain. In the same time era, William Petrie invented the Petri dish so that we could at least look at them. But it wasn't for another 50 years or so that the first antibiotic was described, and that was penicillin by William Fleming. And then it was another 40 years or so before disc diffusion was described, but the zones were not yet measured. People just noticed that a disc impregnated with an antibiotic could inhibit the growth of bacteria on a Petri plate. Of course, what happened very quickly after that was the concept of the minimal inhibitory concentration, or MIC. And what you can see here is that obviously the first MIC testing were done in tubes using serial dilutions of antibiotics, which is why we still test in serial dilutions. And again, the MIC is defined as the minimal concentration of a drug which is able to inhibit the visible growth of microorganisms. So you can see right here in this tube, the MIC would be the first tube. These days, it's almost always done in microtiter plates, but it would be the first tube that did not show any visible growth of bacteria. Now, 1967, when disc diffusion was invented, was also an interesting time in the history of medicine. And I have to take us down to Cape Town in South Africa, where in 1967, Dr. Christian Barnard performed the first human heart transplant. I've had the good fortune of being at the hospital that's there. There is a museum that is set up so that you can really see what actually occurred in this groundbreaking surgery operation that occurred. It was just absolutely amazing. And while you were there, you can actually look at some of the records and the history of what happened to the very first patient. And what you can see on this slide is that from day 14 to 20 post-transplant, a lung infection set in. And of course, the patient was administered penicillin at the time, which of course did not do anything for his infection because a Klebsiella was isolated. And for those of you that know a lot about intrinsic resistance, generally these days Klebsiella can acquire resistance to ampicillin and penicillin so quickly that while not intrinsically resistant, it is resistant so often that it may as well be considered for this. If we look at the patient's history is still, there was some improvement, but by day 16, the pneumonia condition had increased. And what I found very fascinating about this museum is you can actually follow what was given to the patient. And I have circled the antimicrobial agents that the patient was given. First gentamicin, which of course was stopped, and then carbenicillin, which, of course isn't a very simple penicillin. And the first heart transplant patient likely succumbed to something that we see all the time today, a resistant Klebsiella. Unfortunately, in 1967, the laboratory could not give too much susceptibility identification to guide the treatment. But today, of course, we can, and we have CLSI to help us in our description of what information we are giving to the patient. The minimal inhibitory concentration is now standardized. It's defined in CLSI M7, and of course, dysdiffusion is defined in CLSI M2. But the MIC is also defined in an ISO document 20776-1 as a reference or overnight method that of course does take 16 to 20 hours. These days, it's a very well standardized biologic method, but it is still highly influenced by testing conditions. For example, the media, the inoculum, the incubation period, the temperature that's there as well. But there are many standards now to help us in our testing that's there. And MICs still do have a lot of value. They can detect emerging resistance mechanisms. You can see low and high level resistance mechanisms. You can define emerging resistance. For example, when we talk about colistin resistance and plasmid-mediated colistin resistance, that was actually found by people screening organisms for their MICs to colistin. We have defined screening cutoff MIC values for ESBL, so knowing those numbers is actually very important. The phenotypes that we see can infer resistance mechanisms, so not only the interpretation, but interpretative reading for our antibiograms. They can simplify complex resistance patterns. They are essential for surveillance studies, particularly those for regulatory agencies. We always like to keep track of where the MICs for certain drugs and bugs are, so we can see if there's any increase. This has been seen a lot with Streptococcus pneumonia and penicillin. And the MICs do not change over time. The MICs are for a given organism and given testing don't change. The breakpoints may change, and they do change very often in what is defined as resistant or susceptible, but the MICs really don't change. And they're relevant even when using molecular methods that are there. So still something that's very important to us. So now just to kind of go back to our timeline, in 1977 and in 1981, automated identification and susceptibility testing came into play with the Vitek system first commercialized by McDonnell Douglas and the microscan system as well. In 1983, the AS4, a semi-automated plate reader for microscan was developed, and of course it's still in use today. For some of the more modern techniques, DNA sequencing was first described in '77, the polymerase chain reaction in 1983, next-gen sequencing in 2000, of course something that's very important to all of us these days, the use of MALDI-TOF for identification of bacteria and fungi was launched in 2006. And finally, molecular tests really came on the market, so to speak, and were available not for research purposes. And they were the first CLIA wave tests were finally seen in 2014. So we've had kind of a really good past 40 years of, microbiology that's there as well. Of course, leading, to a lot of new tests that have been developed for, again, susceptibility testing of bacteria. And it's very important these days. We have a lot of priority pathogens. I'm looking here at the World Health Organization list. They've defined as priority 1, critical, priority 2, high, and priority 3, medium. What we notice, of course, about the critical list is that it relates to carbapenem resistance, either in the Enterobacteriaceae or the Enterobacterales, for Pseudomonas aeruginosa or Acinetobacter baumannii. But of course, ESBL producers are still very, very important. So the priority 1 pathogens are really defined by those that are resistant to a lot of the higher level beta-lactam and carbapenem drugs. And what you'll see in the priority 2 lists that are high, vancomycin-resistant Enterococcus faecium, methicillin-resistant staph, and vancomycin intermediate and resistant staphylococci. Of concern these days is fluoroquinolone resistant salmonella. And then finally, those that are medium priority may be important in your area, strep pneumo that's not susceptible to penicillin, ampicillin or amoxiclav resistant Haemophilus influenza, and finally fluoroquinolone resistant Shigella. Today, we're just going to really focus more on the critical pathogens as well and spend some time with methicillin-resistant Staph aureus. One of these days, I'd like to just spend some time and talk a lot about fluoroquinolone resistance, particularly in the gram-negatives, as the breakpoints have been changed very recently. A lot of really important work has been done at looking at fluoroquinolone resistant mechanisms in the gram-negative bacteria as well. So why we focus on carbapenems and the beta-lactams. The commonly used beta-lactams, really, if you look at any commercial susceptibility test system, like a microscan gram-negative panel or a Vitek card, the beta-lactams make up really half of what are tested these days on a gram-negative panel. And what I have on the screen are from left to right, from what I would call the simple beta-lactams or the penicillins, to the penicillinase stable penicillins, to the beta-lactam beta-lactamase inhibitor combinations. I have the classical ones listed here: amoxicillin clavulanic acid, ticarcillin clavulanic acid, of course, piperacillin tazobactam, which is likely a mainstay of testing and use in many hospitals in the world, and amsulbactam, the first generation cephalosporins, cephamycins, monobactams, and again, the carbapenems. The cephalosporins, of course, are divided into first, second, third, and cefepime considered a fourth generation cephalosporin. And of course, these are considered the cephalosporins. These down here, the extended spectrum cephalosporins, and finally the carbapenems. What is critical, of course, is that the carbapenems can, of course, give resistance to all of the beta-lactams that are there, as can the extended spectrum beta-lactamases. So it's very important to be able to detect resistance. So looking first at the Enterobacterales and carbapenem resistance, really any of the Enterobacterales can become resistant to a carbapenem in 2 ways. The first, and one of the ones that we'll deal with today, are the production of a carbapenemase enzyme. And these, of course, are referred to under various acronyms, generally CP or carbapenemase producing CREs, CPEs as well. The Enterobacterales have acquired genes quite easily for many diverse enzymes for carbapenem resistance. Most of these are plasmid mediated, So these are easily transferable between strains, species, between genera, E. coli and Klebsiella, patients, and of course facilities. If a patient leaves one facility with carbapenemase producing organism and goes to another facility, it can be transferred quite easily to those to that facility as well. The second mechanism of resistance is that of a cephalosporinase combined with a porin loss. So essentially two mechanisms of resistance. And what occurs here is generally either an AMP-C beta-lactamase or an ESBL. Some of those have low-level carbapenemase activity, and coupled with a porin loss in the bacteria, it limits entry of the carbapenem into the cell. Now, because there's two resistance patterns here, these are not as easily transferable between strains, but still obviously very, very important to the patient. The level of these depends upon your geographic area. Now, where I am talking to you from is in Northern California, in Sacramento. I'm about an hour north of the San Francisco Bay Area. And there have been a number of studies that have been done by Stanford, which is also in the Bay Area as well. And in the Bay Area, this cephalosporinase combined with a porin loss is probably a quarter of the carbapenem resistance that is seen. That is certainly not true, as we're going to see very shortly, in all areas. So our goal for the microbiology lab, is first to detect resistance, because that's what is important for the patient, and then as needed to distinguish between mechanisms of resistance. What we're going to see is that is going to become more and more important as some of our newer drugs are designed for different classes of carbapenemases. So, and then in different areas where you live, it just probably doesn't matter, because those are the organisms that you're going to be seeing, those that produce the carbapenemases. So this is what we're going to focus on as well. Now, the acquired carbapenemases are generally divided into those classes. These are the molecular classes of beta-lactamases. And the carbapenemases are in molecular class A, B, and D. The class A, the example types, are the serine beta-lactamases, and of course the most common example are the KPCs, the Klebsiella pneumonia carbapenemases. They're the largest number. They're almost always on a plasmid in the Enterobacterales. Most of them can be inhibited by clavulanic acid in testing, and that of course forms the basis for the ESBL testing, the MIC of the to the single drug as compared to the MIC of the drug with clavulanic acid. The class B are the metallobeta-lactamases, and of course these are characterized by the most common ones, NDM, VIM, or IMP. And what you can also see that in testing, those can be inhibited by EDTA, and that forms the basis of some tests that are there as well. The enzymes in themselves for the class B do not hydrolyze as trienam, but the organisms that produce these beta-lactamases generally are resistant to as trienam for separate reasons as well. And finally, the class D, the OXA enzymes as well, with the most common occurrences in Kleb-pneumo being the OXA48 and the OXA48-like. These OXA enzymes we'll look at shortly, because these are very common in Acinetobacter and in Pseudomonas and some of the other non-fermenters. So just to kind of take a quick look, I have to thank Dr. Patrice Nordman for these slides. If you just looked at 2007, which really isn't that long ago, it's less than 15 years ago, and looking at the class A or the KPCs, there were a couple of hotspots. One of the hotspots was in the United States. They were endemic in some areas in the Middle East. You could see them in some other areas, but honestly, they weren't that common. If you just fast forward 10 years, what you can see is that they're very common in almost every area of the world. In the United States, we still have a lot of our hotspots that are there, particularly in the New York and the East Coast area. They're common in certain areas in the Mediterranean area, and they're common certainly in China as well, that is there. But even if you fast forward to the year 2021, and again, the same slide courtesy of Dr. Nordman, you can still see that it's a common problem in North America and areas around the Mediterranean, and certainly in South Africa and in China as well. This is kind of what I would say if we were having the Olympics. You know, the KPC enzymes are still really ours in the United States, but certainly they are common worldwide. You know, what I think the pandemic has shown us to everybody is how quickly different organisms can spread. But those of us in microbiology knew this a long time ago, just from the spread of ESBL producers and carbapenemase producers. But again, they are endemic in certain areas, but that does not mean that you cannot find them any place that testing is done. To look at the class Bs, the metallo-beta-lactamases, this of course was the seminal study that was done where NDM, the metallo-beta-lactamases, were seen and were described first in a joint study that was done in India as well as in the United Kingdom. And of course, the reason they were, discovered there were patients were going back and forth due to medical tourism. But they found them in studies both in Chennai, in Haryana, and in all different sites in India and Pakistan, in E. coli, and of course in Klebsiella pneumonia. And what made everyone so concerned about this was what you can see here on the left are the drugs that were tested with, of course, the carbapenems, piptazo, the extended spectrum cephalosporins. In the United Kingdom, in Chennai, and in the state, the Haryana state as well, what you can see is the MICs, these were significantly resistant organisms. The proportion susceptible to a number of different beta-lactam drugs was 0 to very little. And the only drugs that really had an effect on them were tigecycline or the drug of last resort, or colistin. So certainly the definition of multi-drug resistance seen with these metallo-beta-lactamases as well. And these days, what you can still see is these NDM producers are still, of course, very endemic in India and Pakistan and in the areas surrounding those two countries as well. But you see them everywhere, certainly in Africa, in South Africa, in China. We don't see that much of them in the United States. A fair number of them are seen in areas around South America. We certainly see them, but they're not nearly as common in the United States. We, of course, have the KPCs that are there. Looking also now at the class D carbapenemases in Klebsiella pneumonia and E. coli, again, you can see the endemic area around the areas of the Mediterranean Sea, certainly still in India, and being seen more and more in Europe and elsewhere. I think that with a lot of these organisms, the plasmids, are in a certain area as well. They can be spread quite easily, but again, what you see in a certain area generally are just certain plasmid, mediation types as well that's there. So again, we talked about the goal to detect is first, of course, to detect that carbapenem resistance, and then is needed to distinguish the two mechanisms. And there are some studies in the literature. I'll look at both KPCs and then another study that looks at different types of these carbapenemases. And this was a study that was done in the United States several years ago that looked at 46 confirmed KPC producers by a variety of commercial susceptibility testing methods. I'm going to focus right here on the top on the CLSI meropenem interpretive criteria. That's today's CLSI meropenem interpretive criteria. These are the old US FDA breakpoints, which certainly no longer apply. But what you can see here is that they tested these organisms, and what they were doing, we're looking at whether or not there were very major, major, or minor errors. To remind ourselves, what a very major error is that the organism is truly resistant, and the test method calls it susceptible. This is a very bad thing. Major errors is the opposite way. The organism is susceptible, and your test method calls it resistant. So not that much of a patient safety issue, but certainly limits the drugs that can be given to that patient. And then minor errors are anything that has to do with intermediate as well. And what you can see here is that the microscan system had 0 very major errors. And we've taken a very good look at all of our products to make sure that we are able to detect carbapenem resistance. And I think the accuracy of the product of the microscan system has been shown in a number of different studies. It just cited the Bulick one as well. That was for KPCs. In South Africa, of course, they see more NDMs and OXA48. And this was a study that was done by Dr. Olga Perovic, looking at carbapenem non-susceptible isolates at the Antimicrobial Resistance Lab in South Africa. What you can see, of course, is what, of course, many of us see with carbapenem resistance. The numbers just, went up and up and up. And what she showed that with ertapenem and good specificity with the other carbapenems that were there, you can look at and see the detection, not only detect carbapenem resistance, but detect some other carbapenemase producing Enterobacterales. And of course, those that are in South Africa tend to be NDMs and OXAs that are there as well. Now, CLSI has described a number of confirmatory tests for carbapenemases. I'm going to talk briefly about some of these. We used to recommend the modified Hodge test as well. That's no longer recommended. The first is some version of the CarbaNP test. CLSI describes one that anyone can do. There are now commercial versions of this CarbaNP test that really have good accuracy as well. The CarbaNP test is very good for Enterobacteriaceae and for Pseudomonas aeruginosa. CLSI has also described what is the MCIM test that is good for Enterobacteriales that are not susceptible to one or more carbapenems. These are very easy to do. There's no special reagents other than a meropenem disc that's there. And then, of course, there are a lot of molecular assays that are there that certainly go ahead and determine the type of carbapenemase. There are advantages and disadvantages to each. Of course, the molecular system is accurate for what it detects. It does take special reagents and equipment specific to the targeted gene, and it only, of course, will detect the known genes only for that. I'd like to just kind of take a brief look at the MCIM test, and because it is, of course, described in CLSI, it's a very easy test to do. And what you are looking at is the ability of a test colony when it's added to broth with a meropenem disc. Of course, the test colony is producing a carbapenemase. It's going to hydrolyze meropenem, and there won't be any meropenem left in the disc. So you incubate that suspension, then you inoculate a Mueller-Hinton agar with E. coli 25922, which is, of course, very susceptible to meropenem. You put the meropenem disc, which may or may not still have meropenem in it, on a Mueller-Hinton agar plate that has been spread with your E. coli line. You incubate overnight and just then look for the zones. If you still have meropenem left, and you can see this right here in the NEG-QC, this is BAA 1706, but what you'll see right here is that you still have meropenem left, The E. coli that's on the lawn will not be able to grow near the meropenem disc. Of course, if your organism that you originally started with is producing a carbapenemase, there's no meropenem left in the disc. And you can see this right up here in the right with the positive QC that's there as well. So again, a very simple test can be done by anyone, including laboratories that might be resource limited as well. You can also, and CLSI has described an inhibitory enhanced ECIM test that can differentiate metallo from serine beta-lactamases. You might recall that I said that EDTA, some of the metallo-beta-lactamases are susceptible to EDTA. What you can do is simply add that to when you EDTA, when you resuspend the test organism in the broth, and you can really then tell the difference between a metallo-beta-lactamase and a serine beta-lactamase. Of course, that can also be done with molecular tests, but again, a very simple test to use. I'm going to go on to ESBL producing organisms and then come back to the importance of why we want to know the classes. We may think that, of course, carbapenem resistance is important, but honestly, ESBLs are still the scourge of everywhere. You can see, of course, right here that it's a worldwide problem. It was a worldwide problem before COVID, perhaps just not recognized, but a worldwide problem. And you can see that in different areas, it's up to 60% of the organisms are producing ESBLs in different areas that are there. And again, third generation cephalosporins that are there. And the ESBLs, those are very important. A study that was done, length of care, and we know this, the cost of care and the length of stay is much greater in the ESBL producing group than those that aren't producing ESBLs. And they're very important in urinary tract infections, which can, of course, lead to urosepsis as well that's there. Again, just looking at some of the detection mechanisms for this ESBLs, and in particularly the CTXM type of ESBLs. This is the one that's spread to all continents and constitute a major health risk. In a study that was done by Dr. Jim Jorgensen at the University of Texas, he looked at the Microscan ESBL Plus panel, which has extended dilutions of the single antimicrobial agent as well as the clav acid containing reagent for detection. He also looked at the neg MIC32 panel, which has a similar streamlined ESBL test that's on pretty much virtually all Microscan panels. And in comparison to reference broth microdilution and disc diffusion, what he found was that the accuracy of the Microscan product was very high. The reason the streamlined version was a little lower than the extended version was that some of these organisms produced MICs that were higher than the highest dilution on the panel, and therefore you really couldn't differentiate between those and an AMP-C. But again, a very good accuracy that is there. Before I finish up with the beta-lactams and the WHO list, I just want to talk briefly about Pseudomonas aeruginosa, cause of very severe hospital-acquired infections, very much antibiotic resistance, almost anything. Susceptibility testing is almost always warranted when isolated. We all know it's a very important pathogen. It's very intrinsically resistant to a lot of different organisms. And Pseudomonas aeruginosa can acquire mutations on a chromosome. It has a lot of chromosomally encoded resistance patterns. It can just as easily require resistance genes from genetic elements as well. And it's, again, very important. Also, the other non-fermenter, Acinetobacter baumannii calcoaceticus, can't really differentiate between the species, so it's generally referred to as Acinetobacter baumannii complex. It's almost always multi-drug resistant, and again, it produces a variety of beta-lactamases, porin mutations, efflux systems, just about anything that you want. Acinetobacter baumannii complex can do. And again, probably 53% of them are multi-drug resistant. So if we go back and look at our acquired carbapenemases, again, the big five that are in the Enterobacteriaceae, but what you can see, of course, the class A, class B, and class D, you can see those again in Pseudomonas aeruginosa. And why it's important to know these is because of some of the new and old antibiotics that are there that we can see. And I've listed some of them down the side, some of the new combination antimicrobial agents, ceftazidime avibactam, meropenem vaporbactam, imipenem relebactam, ceftolozane tazobactam, as well as ravacycline and cefiderocol. What you can see is their activity depends upon the class of carbapenemase producing. So for instance, Avicaz is active against class A and class D, meropenem vaporbactam against class A. For imirel, certainly class A, I think the studies are still out on the class D. But ceftolozane tazobactam is very active against the class D organisms. So these days it's becoming more important than ever to differentiate. And in looking for newer drugs to treat the class B, we see ravacycline and cefiderocol. Certainly tigecycline, an existing drug, and colistin are definitely used to treat these. And of course, there are some newer drugs in development as well, specifically focused on class B and class D as well. So again, the Avicaz, the ceftazidime avibactam, I have some facts that are listed right here for intra-abdominal infections, urinary tract infections, and pneumonia. Effective against gram-negative, including Pseudomonas aeruginosa, so a very important drug. For meropenem vaporbactam, a complicated urinary tract infections against pyelonephritis. It's also effective against gram-negative, the class A carbapenemases, and Pseudomonas aeruginosa. Imipenem relebactam, another new drug, again, effective against urinary tract infections, complicated, not simple urinary tract infections. Of course, it could be used there, but people don't want to do that as well. And again, also effective against Pseudomonas aeruginosa that's there. And again, imiprel, effective against both class A and class D carbapenemases. I think we are seeing a lot of new drugs. That's very good. I'm happy that we're seeing a lot of combination new drugs and new drugs in development for that as well. And again, ceftolozane tazobactam is the one that's really important for the class D carbapenemases, but not effective against class A. Finally, ravacycline, a synthetic tetracycline similar to tigecycline, both IV and oral, so very attractive for antimicrobial stewardship. And this again is very effective against all of the classes of carbapenemase producers, but not effective against Pseudomonas aeruginosa. So I want to just take a few minutes to talk briefly about colistin, the drug of last choice. Again, colistin resistance in the gram-negative bacterial cell wall, which is complicated by, a lot of membranes. Colistin acts by binding the lipopolysaccharide, and resistance occurs when the lipid A component of the LPS is modified. What we've seen a lot, we used to see a lot of intrinsic resistance. What we see these days is more and more plasmid-mediated colistin resistance. MCR1 and MCR2 have been described. You can see that these are, again, found worldwide, isolated from humans, isolated in animals used for food, isolated from food, isolated from the environment. They were really found in animals used for food initially, but have certainly spread everywhere. And colistin resistance is, again, very important to detect. There's a joint CLSI joint working group that has given testing recommendations for both CLSI and ISO. Again, cation-adjusted Mueller-Hinton broth without additives. The trays need to be made of polystyrene. and reference susceptibility testing by other methods cannot really be recommended. So all commercial tests, including microscan, have to be compared to this reference method, and people, take very great pains to do that as well. One of the important things is that the CLSI testing recommendations for colistin have changed with CLSI this year. The 31st edition changed all of the susceptible to intermediate breakpoints. So no susceptible breakpoints are listed, but only intermediate, and a lot of dosing regimens are listed as well. And again, that is true for the Enterobacterales, for Pseudomonas aeruginosa and Acinetobacter baumannii. And the dysdiffusion breakpoints were, of course, eliminated because this is something that can't really be tested using dysdiffusion. Again, only the disc elution test has been described by CLSI. For MCR1 detection, there's been false susceptibility shown with VITEC2. Should not use e-test because it's a because it's an auger type method as well. The microscan panel performance is actually quite good with detection of the MCR1 isolates. And in the US MCR1 colistin-resistant E. coli, we've since tested a little bit fewer more of these. What we've seen is that we can also accurately detect colistin resistance. There has been a study done by the US CDC. Colistin is on all microscan panels for gram-negative identification purposes at 4 micrograms per mil. And the CDC did a study that also shown you could detect colistin resistance on microscan panels very accurately for the Enterobacterales as well as Pseudomonas aeruginosa. They also tested Acinetobacter. Speaking from microscan, we have a contraindication for that, but you can very accurately detect colistin resistance in Enterobacterales and in the Pseudomonas aeruginosa as well. I'm going to move pretty quickly to Staph. I see what my time is, and I do want to talk a little bit about Staph aureus. Staph MRSA has been with us. It's depending upon where you are. I also always encourage people to go to this website, the Center for Disease Dynamics, Economics, and Policy. You can just plug in your area. You can see what the percentage of resistance are. And of course, again, you can see it's very high in the United States. Again, what we've seen is a lot of general and historical types of resistance to beta-lactam drugs with staph. The staph that are susceptible to penicillin these days are very few and far in between. Most of them are resistant due to the altered cell wall PBP encoded by the MEK gene, which confers resistance to all beta-lactam drugs. So if you are an MRSA, you really cannot use any beta-lactam drugs. What we know now is that cefoxitin is better able to detect MEK-mediated resistance in Staph aureus. However, the detection of MRSA is becoming more variable because MEK-C has been described, which is oxacillin susceptible and cefoxitin resistant, and a Staph pseudointermedius and other staphylococci other than Staph aureus are generally more oxacillin resistant, but not cefoxitin resistant. So you really need to look at both that's there. MEK-C again was emerged really first in Denmark as well. Most of the molecular tests these days also test for MEK-C as well. But their study that they shown that was originally done with MEK-C looked at commercial MEK-A methods, which were of course negative. The chromogenic augurs varied by type. PBP2A agglutination was negative because it was looking for MEK-A. And both microscan and VITEC did very well at detecting this if you looked at the cefoxitin screen that's on the well that's there. Again, what we do see though are a lot of increasing variability. There's reported isolates everywhere of staph that are either oxacillin resistant or cefoxitin resistant, or cefoxitin dysdiffusion, or they're positive, or they're for MEK-A or MEK-C, or they're positive for PBT2A. Regardless, if the method is resistant, it's recommended that it gets reported by as oxacillin resistant. And what you've seen in some of the literature is a description of difficult to detect Staph aureus, MEK positive isolates that are associated with oxacillin and false susceptible results, leading you, I don't know that they're false susceptible, they're just not being detected by phenotypic MICs, but they are being detected by molecular systems. And it's not just these. In CLSI, there are both phenotypic and genotypic methods that are described. And in Appendix H in CLSI, there's a lot of information that's there about what happens if it's positive by oxacillin or cefoxitin, negative by molecular, or the other way around. So, kind of a common occurrence. I think the bottom line is that our Staph aureus these days, like every other bacteria, are just becoming more and more variable. So important to be looking for MRSA that's there. I'm going to talk quickly about antibiotic stewardship. I think it's very important in all countries that are there, that are around, that we really focus on antibiotic stewardship so that we don't use inappropriate and unnecessary use of antibiotics in people. I think a lot of this kind of went a little bit by the wayside during the pandemic because there were a lot of empiric uses of antibiotics for people with COVID appropriately. They wanted to treat the patient. But I think it's something that we really need to work on as well. This US CDC has a lot of good information to fight resistance, which of course include tracking as well. And again, there are a lot of information that is there about every time antibiotics are prescribed to be ordering cultures, to make sure the indication, dose, and expected duration are specified in the record, and to reassess within 48 hours. And a lot of specifics for urinary tract infections, where of course antibiotics are a lot of times prescribed empirically for uncomplicated UTIs. Information about pneumonia, apparently, to represent that the symptoms truly represent pneumonia, and information about MRSA and not using vancomycin for methicillin susceptible staph. The roles of the microbiologists here are to really look at antibiotic resistance rates, and there's a new version of CLSI M39 that's there, to monitor resistance rates, to look for our specimen quality and orders for AST, and most importantly, to relay critical results quickly. This is some schematic of something we all do. We have a positive blood culture, we do a gram stain, we let the physician know what it is. I'm using Staph aureus, MEK. Almost all of us can get rapid identification methods from MALDI for susceptibility, and we can give the information to the physician that Staph aureus, if it's MEK positive for an MRSA, and then we can follow up with an appropriate susceptibility test method that's there. So again, important in antibiotic stewardship that is there as well. So I'm going to finish quickly with immunocompromised hosts acquiring a fungal infection. Of course, an immunocompromised hosts are very susceptible to fungal infections. We've seen a lot of rise of those, particularly with, unfortunately, some of our patients that have COVID. This is actually a tree that illustrates that concept. This tree had bark disease, so it had an underlying infection that was unrelated to a fungal infection, and it acquired a fungus disease. In this particular case, you can see these beautiful polypores, but again, is a principle that's there. Both molds and yeasts are, again, considered fungi. And what we're seeing more and more these days is fluconazole-resistant Candida. Candida can do everything in acute or chronic, superficial or disseminated disease, frequently but not always Candida albicans. It can take advantage of the host immune status. It's really an all-purpose pathogen that's there. What we were seeing is that when I started first doing mycology, the majority of yeast used to be Candida albicans. What you can see from this study that was there is the majority of yeast these days are not Candida albicans, but Candida glabrata, Parapsilosis tropicalis, and other Candida species that are there. And again, it's very, very important that we know the yeasts that are there as well. These days, we have a new fungal pathogen, Candida auris, which is resistant to fluconazole and could be multi-drug resistant. Unlike other candidas where you kind of had to get your own, you had your own, Candida auris can be transmitted human to human. It's routinely misidentified when using most traditional biochemical methods, DNA sequencing and MALDI-TOF, and using certain databases can identify it as well. A lot of them are also identified as Candida hamulonii and Duobus hamulonii. It may be the second most common yeast isolated, and there are a lot of cases these days, so I think it's very, very important to do. Finally, I just want to close out that antibiotic resistance is a pandemic that's been here for a while. We've all looked at charts these days for COVID. I hope to show some of the charts for antimicrobial resistance that are there. And I hope that, again, it's not our pandemic. It's up to us as microbiologists to be very aware of antibiotic resistance so that we can make sure that in an age of globalization, where we can look at shipping lanes by GPS traffic. You know, it's a global arena these days, and that was shown. It's been shown so well in the past year and a half as well with COVID. That again, it has been shown with antimicrobial resistance. Just to kind of show a few of the places that I've had the fortune to be in over the past few years. So as a traveler, I probably contributed to the global rate, but South Africa, Hawaii, China, as well as Italy as well. It's truly a global world, and we are all in this together. So with that, I'd like to thank you. And again, I'm happy to take questions. I realize we just have a few minutes for those, so I'm happy to answer any questions that are there. And again, thank you to everyone.
Thank you so much, Dr. Zimmer, for the very informative presentation and also all the shared informations during this microbiology webinar. So now on the question and answers portion of the event, please, as a reminder, you can still click on the question icon on the right side of your screen in order to submit questions. And we have the first question coming from one of our attendees. It's about the new drug combination, Dr. Zimmer, like ceftazidime, avibactam, meropenem, vavobactam, all the list that you mentioned previously in the table. Is Beckman culture having those drugs in microscan panels?
The answer is yes. So we have released eravacycline, meropenem, vavobactam, ceftazidime, avibactam, ceftolozane, tazobactam. If they are not yet on panels that are in your area, it's a registration issue. We have those developed. The one we are still working on is imipenem relebactam. We do hope to have that on panels quite soon, but all of the rest of them are commercially available on panels today and can get them registered in different areas.
Thank you so much. The other question is coming from also one of the channel partners. It's about the EST yeast panel. What are the plans from Beckman Culture for this specific panel?
Yes, it's something that's obviously near and dear to my heart. We have that in research and development. It's something that I think these days is more and more important. So we hope to be able to release that soon. Again, it's very important to make sure that people can detect resistance to fluconazole, to the echinocandins, and also these days some of the Candida auris are also resistant to amphotericin B. So I think it's very important for people to be able to detect resistance to those. There is a dysdiffusion test described for fluconazole, for CLSI. But again, I think it's very important for MICs, and we're very committed to having that and moving that through our R&D pipeline.
Thank you so much, Dr. Zimmer, for your answer. So basically, those are the two questions that I can see. If there are any other questions, please don't hesitate to submit your questions. Anyway, it's almost the hour, so we can get back to you, of course, by answers through your email addresses if you are sending us any questions on the question icon. So that's all the time that we have for today's webinar. Thank you so much, Dr. Zimmer, for your time. I know that it's quite challenging because you are now local time. It's almost 3 p.m. in the morning. So thank you so much for all the efforts. Thank you so much for all the attendees who spent this hour with us, and for people that take the time to ask questions. Please, when the session will be ended, you'll receive a short message. Just click on it in order to submit or to answer the feedback survey. We are interested by your feedback, so it's very valuable to us. Thank you so much for your time,
Thank
and
you.
have a great day all. Thank you. Bye-bye.